Good morning. This lecture continues genetic testing for large-scale DNA changes. The previous lecture was quite long because I realized that you would not have had most of the information before the TUT on Tuesday. The purpose to not give you all the information was to let you read a little bit more and think about the different concepts. I will, however, discuss this in this lecture. The following techniques can be implemented to study large-scale DNA changes. We have discussed the chromosome karyotype, FISH, QFPCR, array CGH, and SNP arrays. I will go through array CGH and SNP arrays again briefly, but will then discuss multiplex ligation dependent probe analysis, Southern blots, and PCR assays and their applications. Just to recap the differences between an array CGH and a SNP array. In an array CGH, the test sample and a control sample are co-hybridized to back oligo or cDNA probes on an array and analyzed depending on the fluorescence signal, indicating copy loss, a green signal, and copy gain, a red signal. In the case of a SNP array, the test sample is hybridized against a SNP array, indicating two alleles, A and B. This is then scanned and analyzed, detecting copy number imbalances, gains and losses. The following analysis will be seen on a SNP array: the log-R ratio and the B allele frequency. The log-R ratio is defined as the normalized pressure of the two signal intensity for the two alleles of a SNP, which in simple terms is the signal intensity of both spots, and a normal log-R ratio of 0 correspond to a normal diploid copy number. If increased above 0, it indicates a gain, and if it's decreased, it indicates a loss. The B allele frequency, defined as the normalized measure of the allelic intensity ratio of the two different alleles and randomly assigned to the A and the B allele, is calculated as a ratio. Signal intensity for the B allele divided by the signal intensity for the A allele and the signal intensity of the B allele. When using both the log-R ratio and the B allele frequency, copy number changes in the genome can be inferred. A deletion is indicated with a decrease in log-R ratio values and lack of heterozygote values, and a duplication, an increase in log-R ratios values, and splitting of the heterozygote clusters in two in the B allele frequency. Let's use the example of a deletion duplication of chromosome 20. The deletion is indicated with the log-R ratio less than 0 and loss of heterozygosity, whereas the duplication is indicated by an increase of the log-R ratio above 0 and splitting of the heterozygote clusters in two. In this SNP array, it's the results of a male patient showing loss of chromosome 3 and gain of chromosome 8q. The bottom represents the B allele frequency, showing loss of heterozygosity of chromosome 3 and allelic imbalance of chromosome 3 and 8 respectively. Note the X and the Y chromosome, where the SNP array indicates that it's a male individual, and there's a loss of the X chromosome and loss of heterozygosity for obvious reasons. In this plot, I have a profile of a breast tumor sample across the entire chromosome 3. The complexity of genomic aberrations coincided with tumor development and is reflected in various and complex changes in the B allele frequency and log-R ratio, indicated several duplications, deletions, and homozygous deletions. Illumina recommends scanning such samples with CMV partition. However, in some cases, due to the sample complexity and the majority of the genome not being diploid, the log-R ratios may not accurately reflect the true copy number change. In this example, where there is a inverted duplication, deletion of chromosome 8p, you can see the duplication with the clustering of the two heterozygote and the B allele frequency, and the deletion with the loss of heterozygosity. I hope this will give you a better idea how to interpret the log-R ratio and the B allele frequency in a SNP array. In some cases, the phenotype does suggest a specific chromosome location and a specific syndrome associated with large-scale deletions of duplications of that region. The region might be prone to changes in copy numbers, such as in the case of the 22q11.2 deletion syndrome. For large deletions, FISH can still be used, but small changes, other methods should be used. MLPA is a quick and versatile method that can detect copy number changes over a broad range of DNA lengths. It is frequently used to scan intragenic deletions and duplications by monitoring the copy number of exons within the gene. And particularly, it's useful in Duchenne muscular dystrophy due to the fact that there is a high number of deletions and duplications in the case of muscular dystrophy in about 80% of cases. It also complements DNA sequencing to scan for copy number changes. Here is a short video demonstrating MLPA. Please click on the picture. MLPA uses pairs of single-stranded sequences called probes designed to bind to specific exons or other sequences called target sequences, whose relative copy number needs to be determined. The probes are targeted to hybridize to a continuous target DNA sequence, and the two probes need to align to give a single probe complementary to the target. The 5 and 3 prime ends of the probes contains sequences not present in the genome. These sequences is then selectively amplified in a PCR reaction. The amount of amplified probe product is proportional to the number of bound copies of the probe, which then depends on the number of target sequences the probe has to bound to. For instance, in a heterozygous deletion, there is just one copy of the target sequence instead of the two, which means the amount of probe of copies is 1/2 of the normal amount, and the amount of amplified product is therefore proportionally reduced. It can also be used in a multiplex reaction where multiple pair of probes can be used to bind simultaneously to different targeted sequence. The left and the right probes for each targeted sequence will have the same set of unique sequences, and the ligated probes can be amplified by a common set of primers. The stuffer sequences, however, are of different lengths, enabling the amplified probes to physically be separated by capillary gel electrophoresis. I use this method for the investigation of unexplained mental retardation, where probes are designed to cover subtelomeric deletions and duplications, as well as common microdeletions and duplications. In the case of large-scale expansions, such as in dynamic unstable trinucleotide expansions seen in myotonic dystrophy and Fragile X syndrome, other methods such as southern blot hybridization and PCR assays are used. Southern blot hybridization begins with the digestion of genomic DNA by a suitable restriction enzyme or restriction enzyme nuclease. The fragments are therefore separated, or the fragments are thereafter separated according to size by electrophoresis on agarose gels, and thereafter transferred to a nylon membrane. Thereafter, labeled probe from a DNA region of interest is allowed to bind to the denatured DNA. The probe detects restriction fragments from the region of interest, and changes in the DNA on that region may be reflected by changes in the size of the fragments. Southern blot hybridization assays have been used to detect long arrays of tandem repeats in myotonic dystrophy and facioscapulohumeral muscular dystrophy. In type 2 myotonic dystrophy, the arrays can expand to as many as 5,000 CTG repeats. In this southern blot of a BG2 digested genomic DNA sample, fragments were separated according to size on the agarose gel. In the case of 1, it indicates a normal person, but in the case of individual 2 and 3, they will indicate expansions in size due to the increase in numbers of the CTG repeats. You can see here and you can see here. Standard PCR arrays are often used to follow modest expansions in oligonucleotide repeats, such as seen in Huntington's disease and Fragile X syndrome. The method uses an external primer which hybridizes to the flanking sequence of the oligonucleotide repeat, but also a primer that can hybridize to target sequence within the oligonucleotide repeat array, as well as outside of it. This then results in various PCR product sizes, which causes a stuttered pattern on electrophoresis. You can see it starts with a C, but it also hybridizes in the oligonucleotide repeat, and you get different fragment sizes. In this example of a TP PCR electrophoresis profile of the CTG repeat and the DMPK gene in type 1 myotonic dystrophy, we see the horizontal axis is the size of the base pairs, and the vertical axis is the amount of amplification products. This electropherogram shows stutter series of PCR products of about 3 base pairs, periodicity indicating an incremental number of CTG repeats. A normal individual on the top and the patient with type 1 myotonic dystrophy on the bottom. This concludes the lectures on testing for large-scale DNA changes. We will discuss testing of small-scale DNA changes in the next lecture. Thank you.